Welcome back to the channel. Today we are diving into one of the most important powerful tools in modern biology, that is RNA sequencing or RNA-Seq. Imagine being able to take a snapshot of all the genes that are active inside a cell at a given moment. That's exactly what RNA-Seq does. It helps scientists track gene expression, discover new transcripts, and even uncover clues about diseases at the molecular level. In this video, we will explore how RNA sequencing works, why it is so important, and how it's transforming research and medicine. RNA sequencing or RNA-seq is a powerful technique that allows scientists to study the entire transcriptome, all the RNA molecules expressed in a cell or a tissue at a given time. It is widely used to understand gene expression patterns, discover new transcripts, and identify disease-related changes in RNA. RNA sequencing works by converting RNA into complementary DNA or cDNA, then sequencing it using NGS or next generation sequencing platforms. The generated sequence data is aligned to a reference genome or assembled de novo to analyze expression levels. There are certain steps includes in the process of RNA sequencing. The first one is RNA extraction. We have to isolate the total RNA from the cell or the tissue. Then selection or depletion of RNA. Choosing messenger RNA using a poly-A selection or removing ribosomal RNA. Third step is the synthesis of cDNA or complementary DNA, converting RNA into stable complementary DNA. Then we have to do the library preparation, adding adapters and preparing fragments for sequencing. In the sequencing steps, we have to run the prepared library on a NGS platform. Then we have to do the data analysis by mapping reads, quantifying transcripts, and identifying differential expression. Let us look the steps in the picture. First, we have to identify the sample of interest. Then in the second process, total RNA extraction is done with the target enrichment. Then fragmentation, reverse transcribe, and ligate adapters then amplify to make the cDNA library. Then that cDNA will go to the sequencing machine where sequencing will be done, and then the genome mapping will be done, and finally the data processing will be done where differential expression, variant calling, annotation, novel transcript discovery, RNA editing, these things are analyzed. There are certain applications of RNA sequencing. For example, gene expression profiling that compares expression between healthy versus diseased tissue. Novel transcript discovery helps in finding new genes, isoforms, and fusion transcripts. Then, non-coding RNA analysis helps in studying regulatory RNAs such as miRNA or INC RNA. Biomarker discovery helps in identifying gene signatures for diagnosis or treatment. Single-cell RNA-seq analysis helps in revealing gene expression at single-cell levels. There are some advantages in this process. That is, RNA sequencing has high sensitivity and wide dynamic range. It detects both known and novel transcripts and provides quantitative and qualitative data simultaneously. RNA sequencing has revolutionized molecular biology by providing deep insights into how genes are turned on and off in health and diseases. From cancer research to neuroscience, RNA-seq is a key tool for understanding life at the molecular level. So that's all for this video. I hope you understand. Please like, share, and follow my channel for more such videos.